Zhang. I'm a professor of ophthalmology at the State University of New York at Buffalo.
Please tell me about your research on AMD.
Yeah, my research AMD is mainly focused on the protein homeostasis regulation in the retinal pigment epithelial cells, or RPE. We have been studying protein homeostasis for many years, and in aged RPE or the AMD pathogenesis, that protein homeostasis is disturbed. One example we can see is that in the aged retina, there is protein aggregation that in both the photoreceptor layers as well as in the Bruch's membrane that has been reported in the human retinas and human RPE specimens. We are interested in, so what's the mechanism of this protein aggregation in there, and what's the role of that? It has been known that the protein homeostasis is regulated by the signaling pathways of unfolded protein response. So unfolded protein response is a cellular stress response that is activated when the protein homeostasis is disturbed.
How do you feel that the protein aggregation plays a role in AMD?
Actually, we think that the protein, this homeostasis and protein aggregation really plays a role in the development of AMD. And the protein aggregation can actually cause the cytotoxicity to the RPE and also can cause inflammation. So that will also play a role in driving angiogenesis. And so we're interested in why protein aggregation happens in the aged retina, and why and how it actually causes RPE damage, as well as the neovascularization in the choroid.
What are the factors you're looking at in the production of these harmful proteins?
Looking at those environmental factors and the genetic factors. So on one hand, for the environmental factors, we're mainly looking at aging and the cigarette smoke. We know aging and cigarette smoke are the two most significant risk factors for AMD. And for aging, we have looked at their signaling pathways that regulate the protein homeostasis, named unfolded protein response, or the UPR. What we found is that one transcription factor that regulates the UPR is defective in the aged retina. So we used the mouse retinas and the mouse RPE, and we compared the young animals and old animals. And what we found is that the XBP1 level and XBP1 activation are reduced in the aged retinas and RPE.
What are the next steps in your research?
Following that findings, we asked what's the potential role of the XBP1 mediated UPR in the RPE and the retina, and that we deleted the XBP1 in the retina. What we found is that the XBP1 deficient retina are more susceptible to retinal degeneration during aging. Present the retinal degeneration that we see, the reduced retinal thickness and decrease the ganglion cell numbers, which meaning is that so there's a deficiency in this XBP mediated pathway could lead to premature aging in the retinal degeneration.
How do you see that this research can translate into patient care?
We also asked whether we could translate these findings into a patient treatment. And what the first experiment we did is that we overexpress this XBP1. So we express this active form. And we found that overexpressed XBP1 can in fact protect RPE cells. And we exposed the RPE cells to cigarette smoke extract, and then we overexpress this XBP1 in RPE cells. We found overexpression of RPE can in fact protect the RPE cells from cigarette smoke induced damage, that including cell death and their cell barrier integrity disruption. So we overexpress XBP1, you can protect the RPE cells from those damage.
Is there anything else you'd like to add?
So we're now looking at both the genetic and the pharmacological approaches that can activate the protective UPR, including mediated by the X-Pax binding protein 1 or other chaperones that we eventually that we would like to see the development of pharmacological treatment for AMD.